Okay, so first order of business, if you have progress reports, you can quietly get it out and have it on your table to turn in. I believe the word was quietly, though. Quietly. This is not quietly. I do want to go over the fact that Aries is currently updated with your triad grades. I am not going to put anything else in as graded for triad 1. Triad 1 ends tomorrow. However, I put grades in on Wednesday. So I originally said you have to fix everything by Friday. I'm going to extend that to Tuesday. So if there's a test you need to redo, something you need to turn in, anything that needs to be fixed by Tuesday. Okay? I will be here today after school. I will be here Friday after school. I will be here Monday after school, Tuesday at lunch, and Tuesday after school. So you have a good amount of chances to come in and get support if you need help fixing something. Yes, your Unit 2 test is in. I know some of you weren't here to do the retake, so I suggest you get in and do that retake. If you still need more review before you retake, then come in, get review, and do the retake. I did put Unit 2 and Unit 3 syllabus statements in. Unit 2 syllabus statements, you guys got 11 out of 15. And Unit 3, only 7. And I'm going to go over why. Remember that this is a group grade. So your slide might have been beautiful and perfect, but you still have the responsibility of going to others' slides and maybe sending them a comment or an email saying, hey, this could be better, that could be better, here is a suggestion, okay? You guys know that you can @ people in there? I think my third period started doing that, and I didn't realize you could do that, but you could be like, at Sammy, I think your slide needs some images, and then Sammy should get an email, and he'll go, oh, maybe I should go put some images. So you guys can support each other in that way. There were a few people who didn't get credit at all, and I'll show you. Did I show you guys how I grade for credit? And how I go into the history? Okay. So there were a few people who in the history, the name was there like once, but all they did was like click on a box. So I do see in the history like what you do, okay? If you spend more than a minute or 2 minutes on that, I'll see like each minute what you did, okay? So when I click on each minute, I should see a progression of work. Somebody in a different period chose to just highlight a word, and I'm not going to give that credit, right? So you do want to make sure that your contribution is significant enough that I feel good about giving you credit. So if you didn't get credit, that might be why. Looking at this, how you will know what to do is you'll see comments. So if we look down on the slides on the left, were there any comments? Yeah. Unit 2. Actually, Unit 2, there is no comments because your slides looked great. Okay. You followed all the rules. We had great images. Things were good. Things were good. My concern with this was we seemed to maybe forget a bit about the vocab. Okay. One person, I think, was it you, Sammy? Did you get my email? You got to make sure we're putting Unit 2 and Unit 2. So like stippling, field of view, all of this. All of that needs to be in your Unit 2 tab. Okay, so if I'm checking for credit for Unit 2, what I do is I go here to Unit 2. And if I go to history, then I can see, okay, when the last edit was made. So let's say Jaden was in this class and he's looking for credit for Unit 2. When I click on Jaden's name, if I don't see anything highlighted in Unit 2, I'm going to think he didn't do anything for Unit 2. So if Jaden had put his Unit 2 in Unit 1, I'm not going to see it because I'm only looking at Unit 2. Does that make sense? So you want to make sure if you're doing Unit 2 words that you're on the Unit 2 tab. Don't worry, we'll get more used to these things as we go. So that's okay. It's easy to fix. You just copy paste it over, right? So unit 2, I see that we had some words, but there's definitely more words we could put there. Okay. So don't forget about our vocab. That's the reason why I gave us 11 out of 15, because I feel like our vocab needed to be beefed up. So remember, the whole process that I was hoping is you had one slide, right? One slide where you're supposed to work on. Any words that go along with that slide, you got to put in here. Okay. So the person who had the slide with magnification, they were like, oh, that's a good vocab word. Let me go put that on the vocab list. Okay. And remember, this is for our own good when it comes time to study at the end of year 2. Okay. All right. So I will regrade both of these syllabus statements before Wednesday, probably Tuesday night. I'll give you till Tuesday. So as a group, if we fix that vocab, the whole group can then get 15. Okay, yes. I don't know which slide that is. I don't know that we have time to do that right now. But if you want to send me an email and I can double check your history. Okay. If you have a question about if you just got a low grade, that's because the whole class got a low grade. If you got 0 or missing and you think you should have credit, please send me an email and I will go back and recheck it. For unit 3, we have work to do here. Okay. Notice on the left, you'll see that there's comments. If it says like a 1 or a 3, that means there's a comment there. So I have a comment here. That one was unfinished. Okay. So read the comments. That will help you fix it. Okay. And also with a comment, so this one was really good, but if I wanted to comment on this, like, I don't know, maybe I don't like this picture. I can comment and I can say at, and then I could do Sophia, maybe, if she's on this slide. Okay, I can say at Sophia. I'm not sure that this image goes with this syllabus. Okay, and Sophia should get an email from you with your suggestion. Okay, so that could be a really good way for us to help each other. Also, it's not wrong for you to fix this. If you look at this slide and you're like, I don't like that picture, and you have a good reason for it, and you want to fix it or add to it, you could do that too. Okay, I'm assuming we're not going to get any like fights in here over that process. So maybe it's safer to at Sophia and be like, great slide, but this picture could maybe be different. By the way, this picture's fine. I'm just giving it a go. Okay, so you can at each other. That's kind of a cool thing. I didn't know we could do that. So go down, look at your comments. Okay, look at what I said. Fix it, add to it. Okay. Oh, somebody's doing something. Okay, so I will give you guys, I will regrade both of these Tuesday nights. Okay, so we have the potential still to get 15 out of 15 on both of those assignments. And if you got 0 credit, I highly suggest you either do a slide or please go fill in the vocab. Unit 3 vocab could use some work too. Okay, so let's get, I want to go over our unit 2 test. So I'm going to go ahead and pause the recording. Yes, and you can, okie dokie. So we are going back to the beginning of the packet and we're going to fill this out. Okay, so something that we know, oh, I'll just wait. Although I shouldn't have to be waiting. Good time to mention we still don't have a seating chart in here, but should we? Maybe. Okay. So something we know that's different between prokaryotes and eukaryotes is that eukaryotes have these organelles, right? They have more organization. Our Chromebooks are either on this slide or closed. Okay, so we know that they have this kind of compartmentalization to them. For example, the DNA is in a compartment in the eukaryotic cell, in a nucleus, where it was not in the prokaryotes. So what does this mean? What does compartment mean? Go away. I think you're supposed to write this definition down here. That's what I wanted. There's a definition. A separate section of a structure. I'm pretty sure you guys know what a compartment is, but I will tell you at the end of year 2, your brains are going to be so full of stuff. I forget what the easy things are, right? So we just want to make sure. Okay, so a separate section of a container. You can write your own definition, whatever definition makes sense to you. Okay. The advantages. Let's look at this analogy for a second. It's similar to the way a home needs different environments and different rooms. Outfitting each room of your house with all the resources necessary to perform every household duty would be a waste of time, resources, energy, and space. Like imagine if you had a toilet in every single room. Maybe that would be convenient, but is that the best choice? It might be a complete waste of space. And do you really want a toilet in your kitchen? Probably not. Would you need a stove in every single room in your house? Sounds like it'd be handy, but now you're using more space. You're going to be using more energy, especially if it's something that needs to be plugged in and turned on the whole time. So we compartmentalize our rooms to make things more efficient. Okay, so that is kind of the advantage. Okay, so I would just say the advantage, right here in the bold, the advantage is to decrease wasting time, wasting resources, energy, something like that. The advantage is efficiency, okay? To be more efficient with time, resources, energy, and space. Not just time, but resources, energy, and space. It's in that kind of that bold section of that part. Resources, energy, and space. But we're more efficient with our time, energy, resources, and space. It's the same in a cell. Not the whole cell needs to be working on making ATP. If we just have one room in our cell that focuses on making ATP, that process is then more efficient. And we want our cells to be most efficient. Okay, so you're going to get some examples for your next table. You're going to write down 2 examples, which compartment it is and how that compartment is specialized. Okay, so we'll go over multiple examples. You'll pick 2 to write down. So we have 2 examples here. Our compartments are really what we're going to call the organelles, right? Those are the compartments. They concentrate functions into their own interior spaces. This means that regions of the cell can be specialized and much more efficient. Just like we said, that's the advantage. Specialization, efficiency. For example, we have lysosomes. So if you want to choose this one, under compartment of cell, you're going to put lysosome. That is the compartment. And you should already know what a lysosome does because you wrote those notes. But basically, these are like the trash can recycle bins of the cell. I like to think recycle bins because some of what they break apart does get reused in the cell. So they maintain an environment that is more acidic. You don't have to write this whole thing necessarily. Let me read it first. The environment is more acidic than the surrounding cytosol because its enzymes have a lower pH to catalyze reactions. Similarly, pH is regulated within mitochondria, which helps them carry out their function of producing energy. So we actually have 2 compartments here. But the lysosomes, if you're choosing that one, we could say more acidic for enzymes to do their job. More acidic, they're more acidic for their enzymes to do their job, okay? Because how it breaks down things is it has these powerful enzymes. Enzymes are there to break things apart. If you think about your stomach, which is to break apart your food, it's full of enzymes, and those enzymes are breaking things apart. Those enzymes need a more acidic environment. So that's how this compartment is specialized. It specifically has a higher acidity so that those enzymes will work in that compartment. Okay? This is also kind of a protection thing because if for some reason those enzymes get out of the lysosome, we don't want those enzymes digesting our nucleus or our mitochondria, right? So those enzymes, which are probably being made in your rough ER and going through your Golgi, are only going to be functional once they get inside the lysosome, right? Like if you have attack dogs, you want to make sure they're only attacking when you want them to attack, right? You don't let them out when your family comes over because you like your family, probably, maybe, I don't know. Okay, so let's go down to the third one here. Oh, if you chose mitochondria, that was probably not a very good one. It's not super specific. The next one, some proteins require an oxidative environment for proper folding and processing. But the cytosol is generally reductive. That is more of a chemistry term. Therefore, these proteins are produced by ribosomes in the endoplasmic reticulum, which maintains the necessary environment. So if you want to do ER, endoplasmic reticulum, that would be a compartment. You want to do your second example right now. The compartment of the cell is the ER, endoplasmic reticulum. How this is specialized. It is a more oxidative environment. More oxidative for proper folding and processing of proteins. So our ER is our compartment. It is more oxidative, which just kind of has to do with the chemistry of the environment. More oxidative to allow for proper folding and processing of proteins. Because we know that proteins are made in the rough ER. So actually this should probably be RER, rough ER. But in order for those proteins to be properly folded, they need an oxidative environment. You want to know more about that? That's going to be more of a biochem class. It is. And I did take that in college. I think I have my biochem textbook in that. Ooh. What's your overall goal? Healthcare? Oh yeah, you told me that. Nice. All right, we also have proteins are often then transported within the cell through the membrane-bound vesicles. Vesicles would be another compartment. They're not super specialized though, so we won't use that as an example. We have some more examples. If you don't already have your two, you can take from these. The genetic material of eukaryotic cells is compartmentalized within the nucleus. The nucleus is specialized because it has a double membrane, and that double membrane gives it additional protective enclosure of the DNA. As a result, eukaryotic DNA is less susceptible to mutations. It makes sense that we want to keep our DNA super, super safe, right? So it's like having, you know how some of you guys might have like a door and a security gate? I have a security gate. I like it. They exist. Yeah. Mostly I like it because I can leave my door open and get airflow and I don't have to worry about people seeing it. It's mostly my issue, but it's, I mean, I really do like my neighbors actually. So it's more my husband is likes his privacy. I don't care. But it is nice having that security door because you never know. We also have something called peroxisomes. Peroxisomes, which I know wasn't in your notes, and I kind of was like, you should probably add that. They protect a cell by containing and neutralizing dangerous substances. They're kind of similar to lysosomes, but they carry out specific chemical reactions that produce hydrogen peroxide. You guys know hydrogen peroxide is in the brown bottle. You put it on your cuts and wounds sometimes. That's actually toxic to your insides.
I think that's bad for you, no?
Yeah, and actually a lot of first aid, and the last first aid training I went to did say that they don't actually use hydrogen peroxide anymore. I always grew up thinking you put it on there, and if there's bubbles, that means it's working, right? They actually said it could make your wound healing take longer. But it makes sense because it's toxic. If you drink it, that'd be bad. But also maybe kind of why it works, right? Because it's going to kill things. But they also contain enzymes that convert it to harmless oxygen and water. So these peroxisomes are specialized because even though their job is to make toxins, they also have a way to fix those toxins. Also, David, I see a lot of movement. We've been on one slide for a long time now. Let's stay on this one. I'm not even 100% convinced that most of you need the Chromebooks. I understand over here because I'm in your way all the time. But I'm trusting. Why are we still clicking? You're trying to get to this slide? Okay. Just make sure you're on the slide. Okay, so it's specialized because it has a way to deal with the toxins that it creates to protect you. Okay, I wish we had time to do this matching and a way to do this matching, but Nearpods are kind of a thing of the past, for me at least. We already did this. This was your matching. And we have to do this. Yes, yes. State the structural differences between plant and animal. Is a plant cell a eukaryotic cell?
No. No. Yes. Yeah, it is.
Yes. Plant and animal are both eukaryotic. Okay. I should actually get my models down. I'm short and I'm not allowed to be on a chair.
I'll get it.
But if somebody wants to grab those models, those two. We have plant and animal. It would look great. You could definitely stand on a chair because they're pushed back a little bit. But they have a lot of similarities, right? So a plant cell obviously, hopefully, doesn't look like a prokaryotic cell. If you looked at it, you would not think, oh, that's prokaryotic. Okay. Oh, wonderful. Here's our, which one is this one?
The plant. Animal.
Animal.
That's the plant one.
Please. Somebody said they remember this from Mr. Man's class. Yes. Oh, yes. We're friends on Facebook, but she posts almost nothing. But one of her sons had a birthday and she posted like a family photo.
Alvin.
Actually, I don't know which brother it was. They were both in the picture. They're so tall, like even taller than they were. Anyway, she looks like she's doing good. I should check in with her. I don't know. She was so cool. She was one of like, I remember going to Atlanta with her and we had a good time.
Whoa.
What happens in Atlanta? We were at a science convention, actually. But we did also have fun. We were there on St. Patrick's Day. We went out that night. We did go to Waffle House, but it was like 3 in the morning and I was not even hungry at all. And all I wanted to do was sleep. Okay, hopefully, oh, by the way, you probably don't need to write animal cells do not a million times. Please do write animal cells store glycogen. Okay, so see the little parentheses. I think the animal cells do not is kind of obvious, but for the store starch, I want you to know that difference. Okay, so hopefully we know that plant cells have cell walls just like prokaryotes, but they're made out of different stuff. They have plastids. The only plastid that we actually study is the chloroplast. And you guys know that's where what happens? Photosynthesis. Thank you, one person. They have a large central vacuole. That's important for maintaining shape and structure. And turgor pressure, yes. And they store starch as energy. Both of these cells store energy, but they store it in different forms. When we get into unit 12, maybe, we'll talk about the difference between glycogen and starch. Okay, they're both carbohydrates, they both store energy, slightly different molecules. And they have plasmodesmata. That's probably a new word for you. Great vocab word. Let's feature syllabus statement. Plasmodesmata. Look at it here. It's like a little alleyway between two different cells. You can actually see them on the back of the cell. See all these little indents? Those, if another cell was connected right here, it would be an alleyway. So nutrients and things can flow between two cells. Okay? An animal cell doesn't need that because it doesn't have a cell wall. This is specifically like an alleyway between a cell wall. Kind of like a secret.
So they all have identical ones?
The plasmodesmata?
Yeah.
Yeah, I guess because it's really just kind of like if you just bored a hole through a wall. Yeah, it's a very simple structure, but it allows things to move between cells. It is very easy for things to move between animal cells because there's no cell wall. Okay, animal cells. What's different about these guys? They have centrioles. Anybody remember what process that goes along with? Yes, it has to do with splitting of cells. What do we call that process? Mitosis. The centrioles is what grows out of. I like to think of them as like little fingers that attach to your chromosomes. Starts with an S. Something fiber. Spindle. Spindle fibers. Okay.
I wasn't going to get that one off. That was my second question.
I was trying to think like if you're into sewing and you have the spindle, but I don't think we even use that word anymore, really. They have cholesterol in the cell membrane. We'll review that when we get to the cell membrane unit coming soon. And they generally have a fixed shape. Sorry, this is backwards. I'm gonna rewrite this. I'm so sorry to do that to you. More irregular shape. Plant cells generally have a fixed shape and are more regular. Okay. Sorry, I know some of you guys go ahead and if I just screwed you up. And we're also missing something. I want you to put here stores. Add a little bullet point. What is its form of energy? Glycogen. Plants store. What do the plants store instead? Starch. Animals are glycogen. Plants are starch. How can we remember that? I remember this by thinking, what's a very starchy food? Potatoes. Also rice. Dough, yes. Potato, is that a plant or an animal? A plant. So you can remember that. You can say, okay, what stores starch? Well, I know starchy foods are potatoes. So potatoes, starch, plant. Okay. Glycogen, I don't have a trick for that one, but if you know starch, you glycogen, yeah. Okay, so here's your differences between plants and animal cells. Okay, so we kind of went over this yesterday. Basically, we're applying our understanding of these different compartments and these different organelles. and how they help two specific cells function. Your first one was your exocrine glands. Remember, exocrine glands are there to get things out, to exit, okay, to excrete. Yep, pancreas and liver. And if we want to zoom in, here is what the cells kind of look like. So when we look at one cell, notice it's kind of wedge shaped. Yes?
Yeah.
And notice all these dark circles that are going towards one direction. Those are our secretory vesicles. Very good. And those are going to go to getting things out of that cell. Things that we want to excrete, right? We want to excrete sweat. You might not enjoy it, but it's good for you. Okay? You need, there's things in your body needs to excrete to keep you healthy. Okay. So if we zoom in on one of those acinar cells, we can see this real micrograph structure. Okay. So you guys went through this. You looked at what each of the organelles does. Okay, so I'm going to kind of skip past that. We went over this yesterday. Last night's homework was what kind of cell? It was a plant cell. Eukaryotic plant. Palisade. I've heard mesophyll and I've heard mesophyll. I'm okay with either one. Palisade, mesophyll cell. This was last night. So I'm going to go through it quickly. You probably won't have time to write everything. Mesophyll, mesophyll. In biology, there's a lot of, you could say it this or that way. So this is where the site of photosynthesis is very high. So here is just a structure of a leaf. Okay, looking at a leaf cross section. You took a leaf and cut it in half and looked at it on the side. Okay, this is what you would see. We have the mesophyll layer. And we notice that there is a palisade layer and a spongy layer. Okay, we're in the palisade layer. So what does that look like in real life? We zoom in. Oh, first I should read this. These cells contain the largest number of chloroplasts. We know photosynthesis is super important. So this is a very good cell to study. Because what we're going to see is a lot of chloroplasts, because this is the primary site of photosynthesis. Here's what it will look like in real life. They're very rectangular, okay? That is a trend for plant cells. Like we said, they're more regular or fixed shaped. So you're gonna see they're rectangular or usually rectangular, could be squarish. And you can notice that it's different than the spongy, right? The spongy down here is more like a circular in shape rather than the rectangular. Okay. Also, it doesn't look anything like your exocrine gland cell. No. Can you even really see any organelles at all? Yeah. It definitely looks tickly. What we're seeing is the large central vacuole. Something that is oftentimes misconstrued in images is we have a large central vacuole, like right here, this big empty space in this model. Most of the time, it takes up 80 to 90% of the cell. So poor plant cells, all of their organelles are squished to the side. And they have this large central vacuole. So in real life, that's what you're seeing. This large central vacuole is this white space. And all of the organelles are squished in on the side. Okay. No, because that large central vacuole is so important. But like I said, most of the images you'll see will look more like this. Because in order to study, right, we need to see the organelles that are in there. So the drawings kind of shrink the large central vacuole so that you understand it. Technically, yes. Okay. Draw a labeled diagram of a palisade cell from a leaf. You are supposed to do that for homework. Okay. Things you want to show. are these organelles. Okay, like it says here, polyphase cells have additional structures such as ER and Golgi, but they're not emphasized because, for one, they're very difficult to see, and for two, they don't really do anything with photosynthesis. Okay. All right, so again, you write down the organelles. I do want to go through this again. So if you're writing, I need you to pause. Maybe take 1 second reflection of why is this writing not already done? Can I do better with my homework? But either way, pause the writing. And look up here. Because on a test, you might have questions that will show you a real image, a real micrograph. You will have to identify what it is you're looking at. I want to give you the tricks and tips for that. Oops. So that first one, the nucleus, generally, other than in plant cells where you have a giant, large central vacuole, oftentimes will be the largest structure. Okay, it has a clear membrane around and usually has a dark spot, which is the nucleolus. The mitochondria, not always a jelly bean shape. Okay, but oftentimes it is. The best way to identify the mitochondria is by the line in the middle. The matrix is what we call it. Okay, kind of looks like, I don't know, maybe like a mazy kind of shape. Your chloroplast. Again, sometimes they look like this, sometimes they're more squishy. If you have a color image, it will be green, but on your IB test, I think they're black and white. So what you want to notice is what we call the thylakoid. See how there's like these stacks right here, and then there's like lines between them. So you kind of just have to recognize this pattern, okay? And that's going to be a chloroplast. Huh? Yeah, definitely. Looks like a leaf. That will help you remember it's chloroplast. The large central vacuole is easy one. Big white space. Your eyes should be up here, not on a screen. And it should be down. The cell membrane, that's pretty easy. It's just around the outside. Your cell wall, you can see that it's like right here would be your cell membrane, and your cell wall will be thicker, a thicker space on the outside of your cell membrane. So if you were drawing this, you would draw your cell membrane as a thin line, and then you would do a thicker space with your cell wall on the outside, okay, to show that space. All right, can we identify these structures? Big white dot, dot. Large central vacuole. There's other vacuoles, okay? So in the plant cell, you want to say large central. This one, chloroplast, very good. This one, nucleus. This one, I think cell wall, because this one's pointing to the membrane or plasma membrane. Yes, very good. Cool, cool, cool. Okay, you guys are supposed to do these drawings on your own. Okay, again, in here, in terms of labeling, I would expect you to be able to label what? Mitochondria, yep, down here or up here. Chloroplast, nucleus, even the nucleolus, if you know that, label that. And large central vacuole, cell membrane, cell wall. Okay. So make sure you draw that in your notes. You have, I know it's not half a page space, which is what we generally shoot for, but you have maybe a quarter page of space in there. All the little ones, yeah, they might be lysosomes, peroxisomes. So again, you should be able to have a picture that looks similar to this. Okay, so you got to do that at home on your own. Yeah, much smaller compared to chloroplast, which is another misconception. Because in plant cells, I don't know if this one does. This one's, the chloroplasts are a bit larger than the mitochondria. I've seen a lot of pictures where the mitochondria and the chloroplasts are the same size. I'm like, ooh, that's not realistic. But also, we may not have known until recently because our technology is advancing. Okay. What's next? I know. I gotta figure out what I need to do. We're actually almost there, right? I think, let's see, tomorrow we don't have any plans in here other than notes, right? Okay. I think I want to give you a break of homework, except for your syllabus statements. I think tomorrow we can definitely get through page 8 and 9 and hopefully 10. If not, you'll have 10 and 11 for homework. But I think we can get a lot of our notes done tomorrow. That'll be our goal, to finish the notes tomorrow. Then we will have Monday to study, and then you will have your test on Tuesday. Okay? Sound like a plan?
Yeah.
All right. Wonderful. On my little stand over there.